Welcome to your physiology classroom, and today we are going to discuss about action potential. This is a neuron, something we have been studying since quite a long time. We know that these are the dendrites, and this is the cell body, this is the axon, these are the synaptic terminals. This place is called the axon hillock. What are these? These are the Schwann cells. What are Schwann cells? Schwann cells wrap up the axon, forming the myelin sheath. And the gap in between the two myelin sheaths, the gap in between two Schwann cells is known as the node of Ranvier. So with this basic understanding of the structure of a neuron, let us move on to the concept of resting membrane potential. Right now, our neuron is in the resting membrane potential. What do I mean? Let's assume while we are starting our discussion, our neuron is in 0 millivolts. There is this pump. This pump is known as the sodium potassium ATPase pump. It pumps 3 sodium ions outside the cell and takes 2 potassium ions inside the cell. So what is happening? Sodium is increasing outside the cell and potassium is increasing inside the cell. Okay. Now you see 3 sodium ions are being pumped out while only 2 potassium ions are taken in. So, for 3 positive charges going out, only 2 positive charges are coming in. So, the outside is becoming more positive compared to the inside. This reduces our potential from 0 millivolts to around minus 5 millivolts. Now what happens? There are these channels. What are these channels? These are potassium leaky channels. You see, potassium is more inside compared to outside. So down the concentration gradient through these channels, potassium leaks outside. Potassium leaks outside. Now one thing we should know is while inside the cell, potassium is always associated with anions. These anions are too big to travel outside. So when the potassium is leaving, these anions are staying behind, increasing the negative charge inside the cell. So due to the leaving of positive charge and the staying back of free negative charge, our potential reduces from minus 5 millivolts to around minus 90 millivolts. But what happened? These sodiums, as you can notice, are more outside than compared to inside. So what is happening? These sodiums down the concentration gradient are coming back in. So positive charge is coming in. So potential is increasing. But the number of sodium leaky channels, these are sodium leaky channels. These are sodium leaky channels, like these were the potassium leaky channels. So the number of sodium leaky channels are far lesser than the number of potassium leaky channels. So it slightly increases the potential of the membrane, that is from minus 90 millivolts to minus 70 millivolt. And that is our resting membrane potential. Now we should note that this is equal to the equilibrium potential of chloride. What does that mean? We will see soon. Now let us discuss the concept of Gibbs-Donan equilibrium. It says the presence of non-diffusible ions on one side of the cell affects the distribution of fermiont ions. What do I mean? What I mean is, let us assume this is a membrane and these are anions while these are cations. These cations and anions are freely going out and coming in, going out and coming in. Why? Because they can fit through these holes, they can fit in through these pores. That's why they are going out and coming in. But this particular anion is too big to move out. It can't move out through this small hole. So it is staying in this side of the membrane only and can't reach this side. It can't reach this side. That is why what is happening? It is attracting some positive charges near it to maintain the balance. Although these positive charges could have gone to this side, but because this negative charge, big negative charge is here, these positive charges are staying along with it on this side. So what is happening? This is a non-diffusible ion. The presence of it in this side of the membrane is affecting the presence of these fermiont ions on this side of the membrane. These positive charges are fermiont, but still they are staying on this side. So as we can see in this cartoon, what is happening? This this protein is too big to, usually these big non-permeant ions are proteins. Now this is too big to move out. So what is happening? It is attracting these cations while they could have easily traveled. They are being stuck because of him on this side, and that is attracting more water because of the concentration. Now, if you remember, we studied that minus 70 millivolt is the resting membrane potential of a neuron, and that is equal to the equilibrium potential of chloride. What do I mean? This means that at a particular potential, the amount of chloride that will go outside the cell or will cross the membrane to one side will be equal to the amount of chloride that will enter the cell. So at a particular potential where the net movement of chloride is zero, the amount of chloride leaving the cell is equal to the amount of chloride entering the cell. So the net movement is zero, is known as equilibrium potential. Now how to calculate it? Equilibrium potential can be calculated with the help of Nernst equation. Nernst equation. It is given by, equilibrium potential is given by plus minus 61 log base 10 concentration of a particular ion outside divided by concentration of a particular ion inside. So this is the way we calculate it for a single ion. If we have to calculate it for multiple ions, we use the Goldman-Hodgkin-Katz equation, and that is nothing. Just we do the summation. Same thing. 61.5 log. We just do the summation of the relative permeability of that particular ion with the concentration of that particular ion outside, and divide it with the summation of the relative permeability of that particular ion or ions, and multiply it with the concentration inside. That is how we can find out for multiple ions. So now we can move on to our next topic, that is postsynaptic potential. Now right now our membrane is at minus 70 millivolts. Now there are other neurons as well. This is our particular neuron, and there are other neurons as well. They want to talk to our neuron. This they do through postsynaptic potentials. What do I mean? There are two types of neurons right now that are talking to our particular neuron. Some are good neurons. They are trying to excite our neuron. And some are bad neurons. They are trying to inhibit our neuron. So right now our neuron has minus 70 millivolts. Suppose a good neuron, that is excitatory neuron, is talking to it. So our potential will increase a bit. But then a bad neuron talks to it. So it goes down. Again a good neuron talks, it might go up, and again it might go down. And like this it will continue. These are called postsynaptic potentials. Now if a good neuron talks, trying to excite our particular neuron, it is known as excitatory postsynaptic potential, or EPSP. And if a bad neuron talks to our particular neuron, so it is inhibitory. So it is inhibitory postsynaptic potential, also known as IPSP. Now we will discuss the concept of graded electrogenesis. Here we are talking about only good neurons. Two things can happen. Right now our membrane is at minus 70 millivolts. Suppose several neurons, several neurons, several good neurons try to excite our neuron simultaneously. So what will happen? So it will go up from minus 70 millivolts and come around minus 55 millivolts. Or another thing can happen. One particular good neuron is constantly firing once after another time, one after another time, one after another time, and from minus 70 millivolts it is firing. Before it can come down, it's firing again. Before it can come down, it is firing again, and it is reaching minus 55 millivolts. This type of communication, this type of communication where several are firing together, here, as we can see here, it is known as spatial summation. And in the case of this, Where one particular neuron is communicating repeatedly, it is known as temporal summation. And what is summation doing? It is taking our potential from minus 70 millivolts to minus 55 millivolts. This is called graded electrogenesis. So let us read the definition. The local changes in soma and dendrites are called graded potential. They summate to generate action potential at axon hillock and the initial segment. So we saw how the summation can take place. So what happens at the threshold? So minus 55 millivolt is our threshold potential. Now why is it threshold potential? What happens at minus 55 millivolts? At minus 55 millivolts, these channels are called voltage gated sodium channels. These voltage gated sodium channels open up, as we can see, and sodium rushes inside the cell, and the rushing inside of sodium stimulates the opening up of other sodium channels, and more sodium comes in, and this again stimulates the opening of other voltage gated sodium channels. So this is a positive feedback loop. Few voltage gated sodium channels are opening, sodium is rushing in, and that is stimulating the opening up of other voltage gated sodium channels, just to repeat the cycle, and this cycle is known as Hodgkin's cycle. So somehow we have managed to reach minus 55 millivolts, which is our threshold potential. At that, we have seen that there's a rapid influx of sodium ions inside the cell. Since there's a rapid influx from minus 55 millivolts, our potential shoots up to the peak potential. At the peak potential, what happens? The inactivation gates, these are the inactivation gates of our voltage gated sodium channels, close off. And since the inactivations close off, sodium can't enter anymore. And what happens is the potassium channels open up. So potassium starts leaving the cell. Once potassium starts leaving the cell, our potential starts dropping again. But these potassium channels have slow gates, so they take time to close. So instead of stopping at minus 70 millivolts again, it goes down. It goes down again to minus 90 millivolts. And then the work of the sodium potassium channels, the sodium potassium ATPase pump brings it back to the resting membrane potential once again. So what happens? So initially the sodium channels open up and causes depolarization of the cell, that is taking it up to the peak potential. From the peak potential, it is supposed to come back to minus 70 millivolts initially. That is known as repolarization. But that is not exactly what happens, because the potassium channels are slow to close. So it goes below minus 70 millivolts, it goes below the resting membrane potential, and that is known as hyperpolarization. So what are the properties of action potential? The properties of action potential are, first, all or none. Either it is fully conducted or there is no conduction at all. There is nothing in between. So anything above minus 55 millivolts is an action potential, anything below it is not. That's it. There is nothing in between. Then it is non-decremental in nature. Throughout the length of the axon, the intensity of the action potential remains the same. It does not decrease over time. It does not decrease with length of the axon. And it is orthodromic, that it travels only in forward direction and it never travels back. Why, we will see. So the third property is orthodromic. Then we will see the differences between action potential and graded potential. First, graded potential can be both depolarizing as well as hyperpolarizing. We saw that. While as action potential is only depolarizing. In graded potential, there is no concept of threshold, but threshold is very, very important in case of action potential. Then the amount of depolarization or hyperpolarization depends on the strength of stimuli in graded potential. So the all or none phenomena does not work in case of graded potential, but it does work in case of action potential. The strength of the stimuli does not matter. Anything that crosses minus 55 millivolts is an action potential. We learned that. And the effect of graded potential decreases with distance. So it is decremental. But as we saw before, action potential is non-decremental in nature. So what is refractory period? Refractory period. First, there are two types of refractory period. One is absolute refractory period and one is relative refractory period. What is absolute refractory period? The time from which our membrane reaches the threshold to the first one third of repolarization phase is known as the absolute refractory period, and the rest of the repolarization is known as relative refractory period. In the absolute refractory period, no matter how hard, how strong a stimulus you give, no amount of action potential will be conducted. No matter how strong a stimulus you give to the cell, action potential won't be conducted. But in the relative refractory period, there is a chance that if you give a stimulus more than the threshold, a supra threshold stimulus, then an action potential might be conducted. Why? Why is it like this? Because in the absolute refractory period, the inactivation gates of the sodium channels are closed and it will remain closed till repolarization. But in case of relative refractory period, some sodium channels might open. So what happens if you happen to give a suprathreshold stimulus that is more than minus 55 millivolts, then there is a chance of conduction. Now coming to the question, why is this important? Because imagine this is our action and this is our action potential, that is depolarizing this part, then it is going and depolarizing the next part, and then it is further going forward. But why does it not go backward? Because this part is still in refractory period. That is why no amount of stimulus will make it generate an action potential. It will not get depolarized no matter what. That is why always the stimulus moves in forward direction and never back. That's why the conduction is orthodromic, and this orthodromic conduction is maintained by the property of refractory period. Now let us see some regulating factors. First we'll discuss about the ions. First we'll discuss what happens in case of hyponatremia. We saw that sodium contributes in depolarization. So if sodium decreases, the size of the action potential will be small. So if sodium decreases, the size of the action potential will be small. Then coming to potassium. Potassium is the sovereign ruler of resting membrane potential. So in case the potassium levels increase, in case of hyperkalemia, it makes the cell more excitable by making the RMP more positive. So it will start only from here. And in case of hypokalemia, just the opposite. It makes the cell less excitable by taking the resting membrane potential further down. So we have to start from here and then reach minus 55 millivolts. Then last is calcium. In case hypocalcemia occurs, it reduces the threshold potential. It reduces the threshold potential, so it is easier to reach and our neuron is hyperexcitable. Why? Because calcium stabilizes the membrane. Calcium has membrane stabilizing effect. So if we have reduced calcium, the threshold is also reduced and our membrane is hyperexcitable. And in case of hypercalcemia, it decreases excitability by making the membrane more stable. Just the opposite. Then we'll discuss some factors that determine the speed of conduction. First is myelination. A myelinated neuron will definitely carry action potential faster than a non-myelinated one. So what happens? Myelination, if myelination is there, the conduction is faster. In case of thickness, what happens? If the axon diameter increases, this is one axon A and this is one axon B. If the axon diameter increases, what happens? The velocity also increases. Why? Because the longitudinal resistance will decrease. so the local current can spread farther. So if the axon is thicker, the speed of conduction is faster, so thickness is directly proportional to velocity. And last, temperature. Temperature is also directly proportional to velocity, because in lesser temperature, more current is required to generate action potential. So speed of conduction will be lesser. So these are the factors determining speed: myelination, thickness, and temperature. A myelinated nerve fiber will conduct faster, a thicker one will conduct faster, and at higher temperature, conduction will be faster. Some other factors. Some other factors include this local anesthetic. This is a local anesthetic called lidocaine. Lidocaine, and the toxin of this round fish called pufferfish. We all have seen pufferfish, right? This fish secretes a toxin called tetrodotoxin. So this tetrodotoxin secreted by a pufferfish and this local anesthetic called lidocaine, they block the sodium channels. They block the sodium channels, and as a result, you know what happens. And there's this compound called tetraethyl ammonium that blocks the potassium channels. Okay. Last, the concept of biphasic action potential. What happens in a biphasic action potential? In a biphasic action potential, you have two electrodes. You have two electrodes recording from the surface. You have two electrodes recording from the surface. It also shows all or none phenomena and has a refractory period. It has the concept of refractory period. Now in biphasic, why is it biphasic? Because it has one positive peak and one negative peak, because it is recorded at the surface with the help of two electrodes, right? It is recorded at the surface with two electrodes. So two peaks are produced. One is positive and one is negative. Hence it is called biphasic action potential. That ends our discussion on action potentials today. Hope you learned something from this video. If you liked it, then please do like, comment and subscribe. If you have any opinions, please let us know. Thank you.